In the next two sections, we're going to discuss things called graded potentials and action potentials. What these are simply messages that travel along a neuron, and we'll see the difference between these two a little bit later on. You might remember that a resting membrane potential is the state of the electrical charge on the two sides of a plasma membrane when the cell is at rest. That is, when it's not sending a nerve impulse. So when we have a graded potential, there's going to be a change in the resting membrane potential. You'll remember when we talked about the resting membrane potential, we said that the cell has a negative charge on the inside of the cell near the plasma membrane and has a positive charge on the outside of the cell near the plasma membrane. And typically the resting membrane potential of neurons is negative 70 millivolts. And remember, we always refer to the inside of the cell as being the reference point. So when we say that a neuron has a resting membrane potential of negative 70 millivolts, it means that the inside has a negative charge. When we have a graded potential or an action potential, the membrane potential is not going to stay at negative 70. It's going to change from that. And we'll look at how it changes in a little more detail as we get into these two topics of graded potentials and action potentials. Graded potentials are messages that are generated by a stimulus, for instance, a neurotransmitter or a sensory receptor, for instance, detecting touch. So in other words, if a neurotransmitter is being sent from one neuron to another neuron, that neuron that's receiving the neurotransmitter is going to start sending a message, and this message that's traveling down the dendrite or down a cell body is called a graded potential. So graded potentials are messages that are found along dendrites and along cell bodies. Action potentials, on the other hand, are messages that travel down the axon. You also remember that when we talked about muscle tissue, that action potentials also travel along the sarcolemma of muscle fibers. This is that electrical message that travels along the surface of that muscle fiber and will eventually result in the contraction of the muscle fiber. In this section, we're going to take a look at graded potentials, and in a later video, we'll look at action potentials. Graded potentials are small deviations from that resting membrane potential of negative 70 millivolts. Now there's two kinds of graded potentials, hyperpolarization and depolarization. Let's remember that word polarization that we discussed earlier when talking about resting membrane potentials. When we said that there was a difference in the amount of electrical charge on the inside of the cell versus the outside of the cell, then we said that that membrane is polarized. In other words, the word polarized refers to there being a difference in the amount of electrical charge. One kind of graded potential is called hyperpolarization. This is when the membrane becomes even more negative. That is, the inside of the cell will become even more negative than negative 70. It'll be negative 80 or 90 or something like that. Depolarization, on the other hand, is a type of graded potential in which the inside of the cell becomes less negative, or in other words, more positive. The inside of the cell will have a charge of something that's less than negative 70, like negative 60 or negative 50 or something higher than that. Graded potentials are said to be graded, and what this simply means is that they vary in amplitude or size, and this will make more sense as we look at a diagram here in just a second. And those signals will vary in size depending on the strength of the stimulus. In other words, you might have some neurotransmitter stimulating a dendrite to cause the plasma membrane along that dendrite to depolarize and depolarize just a little bit. In other words, it will go up from negative 70 to, let's say, negative 60. On the other hand, if you have a larger amount of neurotransmitter that is stimulating those dendrites, then the graded potential might be even higher. In other words, you might have a depolarization that will cause the inside of the cell to become something like negative 10, for instance. So when we say that these graded potentials are graded, it simply means that they vary in the amount of change that's going to occur in that membrane potential. Graded potentials are also localized, and what that means is that they will travel just a short distance, and we'll say a little more about that a little bit later on. Graded potentials decrease in size as they spread from the location of the stimulus. And again, this will make more sense when we look at a diagram a little bit later on. And as we've already said before, and this is important, graded potentials occur most often in the dendrites and in the cell body of a neuron. In this picture, we can see the difference between hyperpolarization and depolarization. We've got to remember that although we were seeing an entire neuron up here, graded potentials occur only along the dendrites and the cell body. So let's take a look at a hyperpolarization or hyperpolarizing graded potential. If we were to stick an electrode into the cell in a dendrite, let's say for instance, and we zapped it with a neurotransmitter or even with electricity or with touch, and the neurotransmitter was such that it caused what's called hyperpolarization, then we can see that the electrode that's stuck inside of the cell is going to indicate that the inside of the cell is going to become even more negative than it was before. On the other hand, if a different neurotransmitter sent a signal to the dendrite, and this neurotransmitter is designed to cause depolarization, then an electrode that's stuck in the dendrite near where that neurotransmitter is going to touch, that electrode would show that the inside of the cell is going to become more and more positive or less and less negative. Let's now look at a picture that illustrates this relationship between stimulus strength and graded potentials. If we look down here at the bottom, we can see that the y-axis is indicating stimulus strength. So let's suppose that we're going to stimulate a neuron at the dendrites with a certain amount of neurotransmitter. When we do that, as we can see, the membrane potential will go up a little bit. You remember that we can determine this membrane potential by sticking a probe down into the neuron and measuring the amount of electrical charge on the inside of the cell. And so before the stimulation, the inside of the cell registered a negative 70 millivolts. But as we stimulated it, we can see that the inside of the cell became less negative. In other words, there is less of a polarization, there's less of a difference in the amount of charge between the inside and the outside of the cell. So in other words, the number is becoming larger. Or in other words, it's less negative. There's less difference. It's depolarizing. If we stimulate this same neuron with a stronger stimulus, let's say more neurotransmitter, or we touch it harder, then we would cause the inside of the cell membrane to depolarize even greater. In other words, there's even less of a difference between the inside and the outside of the cell. Instead of there being a negative 70, there's, let's say, a negative 60, for instance, difference between the inside and the outside. And if we stimulate this neuron even harder, then the amount of depolarization would be even greater. So again, this is simply illustrating the idea that there is a relationship between the strength of the stimulus and the amount of the graded potential. That is, the amount of change that's going to occur in the electrical charge on the inside of the cell. This diagram is illustrating this idea that the graded potentials will decrease in size, that is, in their amplitude, as they travel away from the source of stimulation. So here we have a neuron, and we're going to stimulate it with another neuron. In other words, we're going to send a neurotransmitter to this second neuron, the one that's blue. And we've got 3 probes stuck in the neuron that's receiving this message. Because this first electrode is relatively close to the source of stimulation, then we can see that the membrane potential will change quite a bit. The inside of the cell will change quite a bit and go all the way up to a -40, for instance. But the electrode that is further away from the source of stimulation is showing that the amount of change that occurs inside of the cell is less. We can see that the membrane potential did not get up very high. It got up to only a -55. And this third electrode is even further away than the first and second one. And we can see that the amount of change inside of the cell became even less than the first two. The inside of the cell became not quite as less negative or more positive compared to the first two. Since graded potentials change in amplitude or size as they move further and further away from the source of stimulation, we say that these graded potentials are decremental. Decremental specifically means that they decrease in their size as we move away from the source of stimulation.